Good morning, students. Today we are going to study the topic about how physicochemical properties are going to affect the drug action. So before we start with the chapter, we will quickly revise about the definition of medichem, what we have seen in last lectures, and how it is being an interdisciplinary thing which involves with all allied branches. So medicinal chemistry is an interdisciplinary science at the intersection of organic chemistry, biochemistry, computational chemistry, pharmacology, and pharmacognosy. To be precise, it is a branch of chemistry which involves with identification, synthesis, development of new drugs that are safe for therapeutic use. So medicinal chemistry studies how chemical structure influences biological action. Now, in this particular thing, we will be always looking around one of the keyword which is known as structural activity relationship. So if we are going to do any of the modifications on the structure, how it is going to influence the biological action. So this particular fundamentals can be explained by studying how the molecular and physical properties are going to influence the kinetics of the drug. So here when I say physicochemical, so it refers to how the functional groups present within a molecule influence its acid-base properties, water solubility, partition coefficient, crystal structure, stereochemistry, and the ability to interact with biological systems, namely the enzymes what we are going to discuss. Now, for designing of an better agent, a relative contribution of each functional group is there, which we call as pharmacophore, and it overall affects the physicochemical properties of the molecules which are to be evaluated. So studies of this type involves modification of the molecule in a systematic fashion, followed by determination of how these changes are going to affect the biological activity, and such studies, we call them as structural activity relationship. Now, if I say structural activity relationship, so we need to understand the science behind the drug discovery and the designing of the new therapeutic molecules, which are based on all the branches what we are going to study. So namely, we need to understand the organic chemistry, biochemistry, even the microbiology, because when we are going to deal with antibiotics, so we need to understand the microbiology, the pathophysiology of the disease, right? So when we start with identifying the targets for drug development, so a drug target is being identified by identification of the disease condition, therefore identification of the lead molecule, which leads to study of the physicochemical properties with the toxicity also. So most of the lead molecules are obtained from natural sources. For example, the plants, glycosides, alkaloids, terpenoids, minerals, vitamins, and few of the marine sources. When we come towards synthetic category, so we have barbiturates, diuretics, anti-inflammatory agents. Then we have in semi-synthetic category also, which is known as antibiotics or few of the antibacterials. So once the drugs are being identified as its origin, they are to be isolated and the lead molecules are then identified by structural conformation. In other words, elucidation, right? So in confirming the structures, we use preliminary test. We perform the spectroscopic characterization, which involves IR, NMR, and then followed by ultraviolet spectroscopy and the elemental analysis, okay? Now, when we need to understand the approaches for these particular kind of drugs, we need to understand few of the drug discovery models which we are going to study. But whatever model we are going to use, we need to understand how the drug is going to act and what properties are being affected for a drug to become an active pharmacological derivative. So we will shift towards the properties which affect the drug action. So herein we are going to study Ferguson's principle. Apart from that, we are going to study about the solubility, degree of ionization, partition coefficient, drug absorption, bioisoterism, distribution, elimination, and stereochemical aspects of drug action. Now, when we are going to study these particular categories of drug, may it be of any of the origin, we need to understand how the drugs are going to affect the systems and what will be the effect, right? Now, here we will come to a definition. The definition is ability of a chemical compound to elicit a pharmacological therapeutic effect is related to the influence of various physical and chemical properties. Together, we are going to term them as physicochemical of the chemical substance on the biomolecules, which are going to interact with physical properties. Physical property of a drug is responsible for its drug action. And when I tell about the chemical properties, the drug reacts extracellularly according to the simple reactions like neutralization, chelation, oxidation, etc. Now, when we talk about these chemical reactions, we need to understand each parameter, and the parameters goes on like this. So physicochemical properties in relation to biological action. So drug action results from interaction of drug molecule with either normal or abnormal physiological processes. So drugs normally interact with targets. So these targets are proteins, enzymes, cell lipids, or pieces of DNA or RNA. So ability of a chemical compound to elicit a pharmacological or a therapeutic effect is related to the influence of its various physical and chemical properties. So the most pharmacologically influential physicochemical properties of organic medicinal agents, OMAs, are solubility, acidity, and basicity and reactivity. Now, this particular slide explains about how an oral dosage form, when it enters into the GI tract, how it ionizes and how it further absorbs and moves into the bloodstream, into the plasma, and thereby it shows its action. Okay. So, a chemical molecule which acts as a drug will elicit its action only when rate determining step is known. It should be hydrophilic in nature, required for hydrophilicity is required for dissociation. It should be lipophilic in nature, which is required for absorption, and it should be distributed into systemic circulation. Now, drug activity can be classified depending on its physicochemical properties, which determines its potency. So we will classify drugs into main two classes here. One is structurally specific and second is structurally non-specific. Now, what are they? So we will compare both and then we will learn the structurally specific and non-specific drugs with examples. So when we talk about structurally specific drugs, these are specific in their action, whereas structurally non-specific are non-specific in their action. Structurally specific drugs are derived from structural similarities of parent molecule. The example which I have quoted is antipsychotic, whereas structurally non-specific drugs are structurally different from their parent molecule, and the examples are local anesthetics. Structurally specific activity of drug is dependent upon factors such as presence or absence of certain functional groups, intramolecular distance, and shape of the molecules, whereas structurally non-specific activity of drug is dependent on physical properties like solubility, partition coefficient, vapor pressure, and presence and absence of some chemical groups. The activity of structurally specific drug is not easily correlated with any physical property, and any small changes in the structure leads to changes in the activity. When we come to structurally non-specific, substances such as alkanes, alkenes, alkynes, alcohols, amides, ethers, ketones, and chlorinated hydrocarbon exhibits narcotic activity, and potency of each substance is related to its partition coefficient. Structurally specific activity is dependent upon interaction of the drug with a cellular receptor, whereas structurally non-specific actions result from accumulation of drug in some vital parts of a cell with lipid characteristic. Structurally specific drugs action is mediated by interaction with receptors, that to specific receptors, whereas structurally non-specific, the action is mediated by interaction with non-binding sites. Now, binding brings favorable changes when it comes to structurally specific drugs. Binding brings non-favorable changes when it is structurally non-specific drugs. Structurally specific drugs act by competitive action, whereas structurally non-specific drugs act by non-competitive action. Structurally specific are governed by chemical properties, whereas non-specific are governed by physical properties. Now, the relative saturation A is always less than 0.01 in structurally specific drugs, whereas in structurally non-specific, the relative saturation A is always 0.0121. And the examples what we are going to write in structurally specific are diuretics, anticholinergics, adrenergics, and in structurally non-specific, the examples are volatile anesthetics and the bacteriocytes. Now, a very important principle what we are going to study is known as Ferguson's principle. So Ferguson was a scientist wherein he proposed a theory which was given by Mayer and Overton, which explained about the mechanism of action of structurally specific and non-specific drugs. Later, this theory was applied to homologous series of compounds which are non-specific in action, basically the volatile anesthetics. So this showed increase in activity of unconsciousness by increasing the geometrical progression of the drug. Biological action is closely related with partition coefficient, vapor pressure, solubility, and surface tension. So drug concentration is determined at active sites, and a parameter known as relative saturation A is expressed for volatile anesthetics, and the equation is A is equal to Pt by P0, where Pt is partial vapor pressure of drug in solution or in gaseous phase, and P0 is the vapor pressure of pure substance, the lead molecule. For non-volatile substances, solubility plays important role. Hence, relative saturation A is equal to St by S0, where St is molar concentration required to produce biological action and S0 is molar solubility of drugs. The relationship between physicochemical properties and drug action, that is the theoretical representation, was given by Mayer and Overton, and the hypothesis states that the higher the partition ratio P, higher is the pharmacological effect. Further, Ferguson gave us the principle wherein concentration of drug in plasma is directly proportional to its activity, and Ferguson constant is expressed represented by X, where X, we have already discussed the formula, Pt by P0 is equal to X. Okay. Now moving further, herein Mayer and Overton gave an example of the local anesthetics, volatile anesthetics which were used. So wherein he mentioned about the summary of all physical properties of the volatile anesthetics, wherein we can see the examples in the graph are halothane, isoflurane, N-fluorine, desflurane, sevoflurane, and how the onset of anesthesia occurs with sufficient molecules of the H which have dissolved in the cell's lipid membranes, and high solubility has affected the potency. Okay. Now, we are going to study the physicochemical properties which are going to affect the drug action, and the enlisted parameters are solubility, partition coefficient, dissociation constant, hydrogen bonding, ionization of drug, redox potential, complexation, surface activity, protein binding, and isoterism. Now we will study one by one the parameter of the properties which is going to affect it. So the first parameter is solubility. Solubility of a substance at a given temperature is defined as the concentration of the dissolved solute which is in equilibrium with the solid solute. So solubility totally depends on nature of solute and solvent as well as temperature, pH and pressure. Solubility of drug is expressed in terms of its affinity or repulsion for either an aqueous or an organic solvent. So atoms and molecules of all organic substances are held together by various types of bonds, and the bonds are hydrogen, dipole, ionic. These forces are involved in solubility because it is the solvent-solvent, solute-solute, solvent-solute interaction which governs the solubility. So these are the methods which are going to improve the solubility of drug. So to improve the solubility, we can modify the structures by altering the structure of molecule. We can use co-solvents, and for example, co-solvents, ethanol, sorbitol, PPG and PEG. By employing use of surfactants and by using the method of complexation. So how important solubility is? So quickly we will see how solubility is going to impart an role in drug action. So solubility concept is important to pharmacists because it governs the preparation of liquid dosage form, and the drug must be in solution before it is absorbed by the body to produce the biological activity. So drug must be in solution form to interact with receptors.